Good morning. Welcome to Biology 1B. My name is Joshua Povich. I'm the course coordinator. I just want to bring a few things to your attention. First is some important dates that we have. So the drop deadline for this course, it's the early drop deadline, and that's January 30th. So it's pretty soon. Make sure that if you mean to drop the course, you do that. We can't do that for you. The next are the three exam dates. Midterm 1 is Friday, February 27th, 6 to 7 p.m. Midterm 2 is Wednesday, April 8th, 6 to 7 p.m. Midterm 3 and the final together are May 11th, 8 to 11 a.m. Please write those down. They're in the syllabus. Read the syllabus. It's posted online on the website on B courses. You should all have access there. But make sure that you know these deadlines, these dates for the exams. We do not offer exams on different days. We don't do makeups. They're on that day, and we can't do anything about that. So please make sure that you don't have travel planned for those days and that you don't have courses that conflict with taking exams on those days. Okay. My contact information is there, Joshua Povich at berkeley.edu. For enrollment questions, issues with enrollment, anything you need there, Brett Boltz is your person. He's bboltz at berkeley.edu, and all that is in the syllabus as well. Is Brett here? Brett was here for... There's Brett right there. So he can help with any enrollment issues. Labs begin next week. The first day of labs is the 26th. There's, if you're interested in switching your labs, there is detailed information on the B courses front page there. Just scroll down toward the bottom and look at what that says for each of those dates. You can find somebody to switch on B courses, and then there's other detail there. But it's very important that you attend the lab that you are signed up for on the first day of labs. So if you haven't been able to switch, you need to attend the lab that you're enrolled in to keep your spot in that section on the first day, okay? Professor Feldman's office hours begin today after lab. Lab instructor office hours begin next week on the 26th, and through that week there's a calendar that shows all that detail. Look at that online calendar for the most up-to-date information on rescheduling or what's happening with that with office hours. They're in room 5045 VLSB, and there should be signage there, but it's on the 5th floor this year, so it might be a little tricky to find, but it's worth your effort to get there. Your first contact for questions in the class should be your lab instructor. You'll meet your lab instructor next week. Because there are a lot of us, it's just most efficient if you talk to them first, and then they can get you to the right person, whether it's Brett or myself. You feel free to send us emails and ask us, but it will probably be fastest if you first talk to your lab instructor. They have a lot of the information that you're looking for. Okay, and finally, we're doing development for the evolution section, and that's ongoing, and we'd like your feedback on that. So we have two surveys that are available. One is at the beginning of the semester, and one is at the end of the semester. These are voluntary surveys, so if you don't feel like participating in the survey, but you do want the extra credit that's associated, there's 1 point extra credit for doing both each of the first survey and the one at the end of the semester, the post-semester survey, 1 point extra credit. So if you do want that credit, but you don't want to participate in the voluntary survey, there is an assignment that you can do, a small written assignment. But we'd really love your participation in the survey, so if you can just take 15 minutes to do that. You'll be getting an email today about that from Professor Goldwasser. Look for that in your email. It's opened today, after you get the email, through just before the first day of lab, so midnight on the 25th. So the time to do that is really as soon as you get that email. If you don't find that email, please look in your spam or your trash folder. It's a mass email, so it might get bumped into that folder before contacting Dr. Goldwasser and saying that you don't have it. But do look for it today. And after it closes on the 25th, we can offer participation in that after that. So if you are interested in that extra point, please go there. Also, if it's only open to students who are 18 years and older. So if you're under 18 years of age, you can do the alternate survey as well. So thank you for your participation. Look forward to seeing you around labs this semester, and have a great semester.
Okay, good. You all set, Joshua? just to remind you that there are no labs or discussion sections this week. So I know that's a question that some of you may have. I'm Louis Feldman, and I'm going to be the first lecturer in this class. Bio 1B is taught by 3 instructors, and instructor for the third section, Dr. George Roderick, is here, and I want to introduce George to you. George is a population biologist. He also studies conservation biology, and like me, he's a member of the College of Natural Resources. His department is ESPM. And you'll be seeing a lot of George and me, and our second lecturer, Dr. Schabel, who will be talking to you about evolution, was unable to be here this morning. Thank you very much, George, for coming in. Now, Bio 1B, as you have seen, is a large class. You all know it. It has 3 instructors. Our intent is to try and make these 3 instructors seem as part of one course so that you don't have to feel you're jumping from one subject to the other, but that there's a continuation in the material that you have, and it moves seamlessly from one instructor to the other. So even though we have 3 instructors, we're going to try to make this a unified whole for you. Now, this class differs from Bio 1A. It differs from many classes that introduct you on the campus because we are going to ask you to conceptualize, to think, not to memorize. This is not a class where memorization is going to serve you well. You're going to have to be able to put 2 things together and come up with a third idea. And this is really important for you to appreciate that this is what we're going to ask you to do. I realize for some of you it's going to be hard, but nevertheless, this is going to be the objective of the exams that you're going to have and of your labs. Now, this class is not graded on a curve. We have taught this class a number of times before, and we know what you can do. We very clearly set our expectations of what you have to do to get an A or a B, and at the end of the class, we would be happy to give all of you A's. The only person you're competing against is yourself. So I want to say to you, we have these expectations. They're very clear, and they are where they're posted, and you will be able to find out from your GSI whether you're able to meet these expectations. So please realize that it's not one where we have a curve and you have to fit yourself on that curve. It's one in which you're competing against yourself, and we want you to demonstrate to us that you're able to grasp the material, which we from previous years know that you should be able to do. Now, I have been up for a couple of hours, and I've had a couple of cups of coffee, and students have asked, could there be some way that you could get us up to speed, because you're obviously going to be here talking at a very fast rate. And so as a mechanism for slowing me down and for helping you to take notes in this class, I write on the board all of those items which I consider that you should transfer to your notes, that should be in your notes. All of those items which I consider to be important. I'm going to tell you a lot of stories, a lot of interesting facts, a lot of anecdotes, and those are items for your own interest. You are not going to be required to take notes on them, although you're welcome to do so. They are to convey to you the miraculous nature of biology, of nature. So keep in mind that when I write on the board, that's a sign for you to take notes, and also it's a way for me to slow down to make sure I'm not overwhelming you with information. If you can't read my handwriting, or you have a short question which we can answer in the lecture, please shout it out or raise your hand, and I'll be glad to address it. If it's a longer question, then wait until office hours or until you see me later in the lab. Now, we have a number of aids to help you, aside from my writing on the board, to do well in this class. The first thing is that for this class, I have outlines. The outlines have all been made available to you on the web, and they indicate what the subject matter of the lectures are going to be, and they also include diagrams, so you don't have to do them again unless you wish to do so. Sometimes I modify the lectures a day or two before, and so the outline may not perfectly match the lecture. But in general, the outlines and the lectures will match each other. We also have a webcast for this class. The webcast is available to all of you, and the webcast we view as being supplemental to coming to lecture. To give you an idea of what a webcast versus coming in person can do, think of when you go to a live performance versus watching something on television. It makes a big difference as far as the impact. You can follow wherever you wish to go in your with your eyes if you are giving a chance to come to lecture versus being a captive of the projectionists. Sometimes the webcast doesn't work. Sometimes the webcast is out of focus. So if you only watch the webcast, if you only depend upon it, you will probably disadvantage yourselves. It's something which is meant as a supplement to help you here in the class. We also have in this class lots of demos. Have some up here in front. Learning is best accomplished by not only using your sense of sight and sense of hearing, but also your sense of smell and touch. And I want to encourage you to come up to the front. This is the first class in this room. We have the material available to you to feel the material, to smell it, sometimes even to taste it. Learning is best accomplished by using as many of your senses as possible. I take a lot of photographs, some of which you'll be seeing today, to try and help you with the lecture, to supplement the material. The photographs are often taken from here on campus, from plants or places you have passed or will pass frequently, and this is meant to reinforce some of the ideas, some of the concepts that we are presenting you in lecture. You also have a large number of illustrative materials in your textbook. The textbook is a very good source of pictures and of clarifications that perhaps I don't have time to talk about here in lecture. So take advantage of the textbook. I will say today at the outset that I do not take exam questions from the textbook. The exam questions are taken only from lecture. There may be a time when the lecture and textbook parallel each other, but I don't go to the textbook and look for exam questions. There's also the opportunity to send me emails. My email address is posted on the top here. I try to answer the emails fairly quickly. If you send a lot of emails to me just before the midterms, I may not be able to get to them. So try to space out your questions so that I have an opportunity to answer them. There are also office hours. You heard Joshua mention that my office hours begin today, and they are as posted to my left to your right on the board here. You would be surprised at how few of you come to office hours, how few of you take advantage of them. The office hours, maybe 50 of this class will come over the regular semester period. The office hours are a chance for me to meet you, but more importantly, for you to have a chance to come in and to talk. You don't have to raise a very theoretical or very high-profile question. You may just want to listen to see what your peers are asking, to make sure that you're understanding what their questions are about. So I want to encourage you to come to office hours. They are a very useful part of this class, but very few of you use them. Office hours are also a chance for me to get to know you well enough, for some of you, for me to write letters of reference, and also for some of you to ask me, if you're interested, or Dr. Roderick or Dr. Schabel, what it was like to get into the fields that we're in, how did we go about it. You don't have to only talk about the class. This is an opportunity for you to explore other aspects of biology. And finally, I want to say to you that I come to all the labs during the portion of the class that I teach. The purpose in coming to the labs is to give you, in a more intimate setting, a chance to talk to me, a one-to-one, to raise questions, to have discussions. I don't come to embarrass you or to put you on the spot. And so when you see me walk into your lab, don't leave the room. Stay there, because I am there to, first of all, find out if there are some questions you have, and secondly, to find out if there are areas that I'm not explaining myself clearly about. So these are sort of the background for this class, our objectives, and the ways that we have to help you, the ways we have to get you interested in plant biology. The first portion of the class is plants and fungi for the first 5 weeks. And the question I want to put out to you in the very beginning is, why do we as biologists here on this campus ask that you have 1/6 of your introductory class in biology devoted to plants and fungi? And I want to give you a few reasons. Many of you can think of other reasons, but just a few to let us have a setting for this work. First of all, the plants, through the process of photosynthesis, are the organisms which produce most of the food that we as humans eat. There are some organisms, very few, which don't use aspects of photosynthesis or the products of photosynthesis for their foods. But most of us get our nutrients from photosynthesis. Secondly, photosynthesis, which is a process predominantly done by the plants, is the process by which oxygen is regenerated in the atmosphere. So these are important aspects just for your daily lives. But as importantly, if any of you are thinking of going on in biology, and you are going to take more advanced courses, you have to know about the organisms. If you want to be an ecologist or a conservation biologist, you have to know about the organisms which make up the biota. And in order to do so, you have to know something about the plants and the fungi. So I want to encourage you to take advantage of the fact that we are here to present to you a small view, a small picture of this very interesting and unusual group of organisms. Some of you are thinking of becoming physicians. If you are thinking of becoming physicians, I want to say to you, as you'll see in today's lectures, many of the diseases which humans have, many of the drugs which have been developed for humans come or are either based on plants or on fungi. So this will be an introduction to some of you with regard to some of the biomedical interests or issues that come up in this class. Many of you are going to be voters, I hope, when you leave school, if you're not already. And there are going to be a number of issues which come to the fore which demand that you have an understanding of plants and biology. And one of the main issues that some of you are familiar with already is genetically modified foods, genetically modified organisms. What are they? Are they something that you are interested in eating? Would you feed genetically modified foods to your kids? These are issues which you're going to have to vote on, and you can't vote on them without having a knowledge. You can't vote on them in an informed way without having a knowledge of these organisms. And finally, what I want to say to you is that on this campus we have a whole department that is devoted to the study of plants, and I have spent my entire career studying plants. What I'd like to do in the short 5-week period I have is to convey to you the interest and the enthusiasm of this group of organisms by this department and by myself. And I hope that some of this will carry across. We realize that most of you are not going to go on in plants, but from talking with students from the past, we hope that we're able to influence you in a way to see things that you would normally walk by outside and not notice, and now gain a greater appreciation for. So for the first 5 weeks of this class, we're going to study plants and fungi. And these are the organisms which will occupy the lectures, and these are the organisms that you will see in the labs. The labs are an important part of this class. It's very important for you to come to the labs prepared. In some cases, we are asked to collect something from outside and bring it to the lab. You will not have known to collect it unless you read the lab beforehand, unless you prepare for the lab. And your GSIs will take note of the fact that you haven't brought in something which is going to be necessary for the lab. So please read the labs ahead of time. Use them as a supplement to actually show you organisms that we're talking about in lecture, organisms that many of you perhaps have never heard of before, but surely many of you have never seen them before either. So take advantage of the labs. Many big universities have cut out labs, and it's because they are a big expense. We're lucky here at Berkeley. We still have the labs, so please take advantage of them and really use them. So I'd like to begin now with our discussion of plants and fungi. And to begin our discussion, I'd like to go back to the 18th century to the great Swedish taxonomist by the name of Linnaeus. Linnaeus, who was Swedish taxonomist. Now, Linnaeus classified organisms into two groups. He said that organisms were either animals or plants. And based on the classification characteristics that I'm going to give you, the first group of organisms which we're going to talk about, the fungi, were placed by Linnaeus into the plants. So let's talk about what some of these characteristics were. He classified animals as organisms which ate, which were apparently eating something, organisms which breathed, organisms which moved, and organisms which had a life cycle which went from a juvenile to an adult. Organisms which had these characteristics, he said, were animals. And this is contrasted with groups, with organisms which he put into the plants in which there was no apparent eating, no apparent breathing, and if there was any breathing, or if there was any moving, the movement was very slow. So he said there was no movement. And finally, he was not able to assign a juvenile and an adult phase to the life cycle of organisms. And if he was unable to do that, he said that it had an undefined life cycle. And organisms having this undefined life cycle, he placed in the plants. So given this characteristics, the first group of organisms that we are going to study, the fungi, were placed in the plants. And it's important for you to realize that the fungi were placed in the plants back in the 18th century. This is a historical placement. So these were the beginnings of the taxonomy, of the placement of organisms into the various groupings, the two large groupings, animals and plants, by Linnaeus. This type of classification scheme persisted up through the 19th century and into the 20th century. And by the beginnings of the 20th century, it was clearly seen that these particular types of classifications were artificial. They were beginning to break down. And new facts were emerging, new science was emerging, new techniques were emerging, which were superseding these particular characteristics that Linnaeus had used to classify organisms. And I'd like to list for you a couple of broad concepts that Linnaeus didn't know about, but which now underlie all of the classification of organisms based upon more modern techniques and more modern understanding of both the earth and biology. And the first new concept, or first new information which I would like to put out for you, is that the earth formed, and this underlies really all life, earth formed 4.3 to 4.5 billion years ago. It's the first important fact that we need to know with regard to understanding the relationships of organisms. The second key fact, which has emerged gradually and since the work of Linnaeus, is that life originated 3.5 to 3.8 billion years ago, the first life on Earth. So it's possible that a period of time of maybe only 400 to 500 million years elapsed between the formation of the Earth and the origins of the first life. A really remarkably short period of time after the Earth was formed. This is in theory. And finally, the third fact which we need to keep in mind, which has influenced our understanding of the relationship of organisms, is that new molecular techniques have allowed comparisons at the DNA level. And by being able to compare the different DNA makeups of the various organisms, we have now developed a different understanding of the taxonomy, of the relationship of organisms. So these particular new concepts or new facts have resulted in the following classification scheme, which is now the current scheme and which is likely to be modified again as new facts come to light. In the current scheme of the relationship of organisms, of the taxonomy of organisms, at its simplest, we have a common ancestor and this common ancestor, let's say, was 3.8 billion years ago. And from this common ancestor, we have radiating a number of branches. We have 3 branches or lineages radiating from this common ancestor. One which gave rise to organisms which you know of as the bacteria. a second lineage from the common ancestor began, and then it branched shortly after coming off the common ancestor into two other branches or lineages. And these two other branches or lineages are the archaea, probably the least familiar of the branches to you, and the third branch, the eukarya. These is our current interpretation or understanding of the taxonomy at the grossest level of all of the organisms which make up the biota. Now, all of my lectures and most of the topics discussed by the other two lecturers will be on the eukarya branch. But as you can see, there are many, many organisms that we are going to be leaving out. We're hardly going to be touching in this part of the lecture. Now, what I'd like to do is to take this eukarya branch, which is the branch that we're going to focus on, and to amplify it, to actually blow it up and to put some mark or demarcations on it. And we'll begin, so this is the eukarya branch. At the base of the branch, we'll have the common ancestor. And then the first thing we tend to ask is, where are we on this branch? And what we can say is that the animals are toward the top of the branch. They have branched off onto their own lineage. Now, where is that first group of organisms that we're going to study today, the fungi, placed on this branch? The fungi would be placed near the top of the branch, very close to where we are. This is the group, in a broad sense, that Linnaeus classified all the organisms into. All of the organisms that he said were fungi, he would have, if he had known about this scheme, put on this portion of the branch. But now, based upon more molecular techniques, we know that many of the organisms that Linnaeus would have grouped with the fungi, in fact, don't belong with the fungi. They aren't related to them. They are other places on the branch. There are still some organisms which are left here, and these are the organisms which are known as the true fungi. But many of the organisms that he originally classified with the fungi have been removed, have been put elsewhere on the branch. And I'd like to give you a couple of them. We're going to see some of these in the lab. The first group, which was removed from the true fungi and placed elsewhere on the branch, is a group which is known as the slime molds. These are extraordinarily interesting creatures. You're going to have a chance to see them in the lab. They are, if we get some rain, very obviously outside, and if you go into the forest. So we'll talk more about them. Another group which was removed is a group which is economically very important, called Stramenopiles. This is a large group, economically important, because it contains a lot of organisms that cause disease. For those of you who are aware of the disease sudden oak death, which is killing many of the oak trees and other plants as well, these organisms that are doing this lethal process are found in the Stramenopiles and have been removed from the true fungi. But what I want to say, and we'll return to this idea in a moment, is that today and on Friday, when we talk about the fungi, we're going to lump all of these groups of organisms together into the group fungi for our discussion. But they are really, it's an artificial grouping. They really are not related, but it makes it easier for us to have some discussion on this point. Let's just add, since we have this branch up here, a few more markers. Let's put the plants on here. And this would be the green plants. Like the fungi, which had a lot of organisms grouped with them, the plants, the green plants, had a lot of organisms grouped with them that are really not related to the green plants. And a couple of them that we're going to see in the lab and discuss later on are the brown algae which were removed from the green plants because they are very different at a molecular level. And another group that we're going to talk about, the diatoms, also originally grouped with the plants and the green algae, have been removed. So as we go through in our discussion of the organisms, we're going to see that they have different places on this eukarya lineage. Very important for us to think about. Now, the organisms which make up this particular branch are part of the biota. And what we are going to do today is to focus on the fungi, and we're going to focus on the entire group. There are a couple of points which I want you to take from this branch. The first point is one which we just mentioned, is that organisms have been, we'll just put it in a very broad sense, moved about on the eukarya branch. And this moving about or replacement of the organisms in different portions of the branch probably is going to continue for a long time as we discover more and more about their molecular biology, about their DNA, how closely related they are. The second point, which we're going to return to at the end of the lecture, is the following, and this is really an important point. Fungi and animals are pretty closely related. We are pretty closely related to each other. We have a common ancestor here, not so very long ago. This common ancestor, as we will see, means that we share a lot of our molecular biology, a lot of our biochemistry, a lot of our genetics as far as DNA with the fungi. An important point. So, number 2 is that animals and fungi are relatively closely related. And we're going to return to this point in a moment. Now let's talk about the fungi. First of all, I want to mention to you that fungi today are still studied at universities and colleges in departments which are either plant biology departments or botany departments. And this is a legacy from Linnaeus. This is because they were originally studied by people who were botanists, and they continue to be studied today in botany or plant biology departments. But this is a historical legacy. They could just as easily be studied in a microbiology department, having nothing to do with plants. So on this campus, the people who study fungi, who are called mycologists, they are in my department, in the plant biology department here on campus. Now let's talk about the fungi and about some of their characteristics. We believe that the fungi evolved about 500 million years ago, and that today there are about 150,000 named or identified species. But it's been estimated that there are 10 times this number yet to be identified. So this is a very fertile field for individuals, and it's a very active field of study of fungal biology. Again, for our discussion today, we're going to look at all of the organisms, some of which have been removed, as fungi, so that we can have a kind of a compact discussion of this group. Fungi come in an amazing variety of color and sizes. You're going to see some of them in the lab, and hopefully I will have time to show you some pictures of them at the end of the lecture. Now, because we're considering such a large group of organisms, and because we have such limited time, I'm going to have to give you generalities about this group. The generalities mean that some of these facts apply to most of these organisms most of the time, but they don't apply to all of the organisms all of the time. So let's give you some generalities to help you unite the fungi into a group. The first generality I want to give you is that fungi are usually filamentous. What does filamentous mean? That is, they are in a filament. So if you could look at them, as you will see in the lab, there are a group of cells all hooked together into a filament. There are some exceptions, some important exceptions. One of the important exceptions, which is not in the filament, and we're going to see this in the lab, are the yeasts, not filamentous. A second characteristic which unites the fungi is that they are not photosynthetic. That is, they are unable to take CO2 and water and use sunlight to manufacture compounds. They are not photosynthetic. Essentially, fungi live in their food. They live in their food. They secrete enzymes. And they then digest the compounds and absorb them. Essentially, the fungi's stomach is outside of their body. That's where all the action goes on. A third characteristic of most of the fungi is that their cell walls contain a compound known as chitin. This is the same compound that you find in the walls of insects, in the body of insects, or in mollusks. So it occurs several times in nature. The fourth generalized characteristic is that the life cycle, which we'll talk about next time, can be both sexual and asexual. And finally, the last generality of many that we could write on the board, but are the ones that we consider important, is that they produce structures known as spores, which are unicellular bodies capable of giving rise to of a new organism. So these are the important facts that I'd like you to keep in mind with regard to trying to unite this very big group of 150,000 organisms. Fungi are the great decomposers of the world. Along with the heterotrophic bacteria, and a few other groups, which we won't mention here, the fungi are able to digest, to decompose almost and probably likely every naturally occurring substance. They have enzymes made to digest and decompose. The activities of the fungi and these other decomposers are as essential for life as are the activities of the photosynthetic organisms. Because only by decomposing organic material do we again then release the minerals and the nutrients back into the soil in order to be reused in making new organisms again. So although you may look at the activities of the fungi as detrimental, they are important to life. Now, a fungus is equipped with a powerful arsenal of enzymes when you look at the fungi as a group. They can, as I said, digest almost anything. It makes, in the life cycle of fungus, it makes no distinction between a dead, fallen tree in the forest and a fence post that we might put up. It can decay both of those. But these qualities, in other words, the enzymes or the proteins that the fungi produce, which in many cases are so detrimental, are also used by humans in many ways. And here are some examples of fungi or fungal activities which we as humans use. Much of the fungal activity that we as humans consider beneficial is involved with food. The production of CO2 in the bread-making industry is a reaction of yeast. The formation of alcohol and alcoholic spirits is a result of the yeast fermenting the alcohol and making the alcohol and using sugars. So here we have an organism that is able, through the production of enzymes, to be able to influence our food industry. The cheese industry is based upon the production of compounds by fungi. In days before refrigeration, it was discovered that if milk was contaminated with fungi, that somehow the milk was preserved. It didn't get destroyed by bacteria growing on it. And that is because the fungi produce compounds, which we'll talk about in a moment, that prevent the bacteria from growing and destroying the dairy products and making the many different types of cheeses that you have experienced. If any of you have gone to a factory where they're making camembert cheese, you might smell ammonia in the air. The ammonia is a byproduct of the fungal digestion of the milk products, and is thought in this way to be a way of the fungus to prevent other organisms from growing near or on the cheese. So a lot of the lore of fungi, a lot of its usefulness is in regard to the food industry. Also, though, a lot of the lore related to fungi is in regard to its producing toxins, which we consume. Every year, if we get some rains, you'll read in the newspaper of people who go out collecting mushrooms in the Berkeley Hills and of people who die. And they usually die, well, you guys have told me a lot about your perspective here. They usually die from eating a fungus known as Amanita or the death angel. This fungus produces a compound, a toxin, which is probably useful in retarding other organisms from eating it, but it is not destroyed when the food is cooked, and when people consume it, within the 24 hours after they consume it, they suffer usually irreparable liver damage, and the only way they can be saved is by having a liver transplant. So fungal toxins are a big concern to humans. Another fungal toxin, which is of concern to some, but not all people, is a toxin known as aflatoxin. This is a toxin that is produced by fungi, which grow on a variety of organisms, a variety of crops. And in the United States, the U.S. Department of Agriculture allows a certain low level of aflatoxin to be present on all organisms. It doesn't seem to interfere with most of our daily lives. But for those of you, for individuals who have had hepatitis B or C, the consumption of any aflatoxin is, through a mechanism that I don't know, able to stimulate cancer formation in the individuals. So people who have had hepatitis B or C have to stay away from even touching organisms or plants which have aflatoxin possibly growing on them. And a common crop that many of you eat, which has a small amount of aflatoxin contamination, is or are peanuts. Peanuts have a large potential for having organisms to produce fungi growing on and produce aflatoxin. So for those of you who have flown on planes, you will get little packets. In the old days, they used to say this packet contains no peanuts. It was not even made in a facility where peanuts were processed, because individuals having this sensitivity, being exposed even to touching something, can stimulate a response in them. Many of the compounds that the fungi produce, which we consider to be toxins, are also very useful for us. One of them that many of you who go into medicine will encounter is a compound known as cyclosporin. Cyclosporin is an anti-rejection drug. It is used to give to people who have problems with transplants, so that the transplants aren't rejected. Probably the most famous compound that has been used and is the basis for a whole industry is penicillin, produced by the fungus Penicillium. This is an antibiotic which the fungus Penicillium produces, and it is probably meant in the life cycle of this organism to prevent bacteria from growing and disturbing this particular organism. We as humans use penicillin to fight bacteria that may invade our bodies. So these are very useful products that fungi produce. Now, fungi also cause a large number of human diseases. For those of you who have had, you may not recall this, diaper rash when you were very young, or may have ringworm, or perhaps have toenail fungus, these are all diseases which come from fungi. For those of you from the Central Valley of California, you probably are familiar with Central Valley with the fevers, that the respiratory fevers which come from fungi in the atmosphere. These are diseases caused by fungi, which in most cases we as humans are able to fight off. We as humans are mostly able to fight off and not have to deal with. But for individuals who have their immune systems compromised, and I'd like to just mention people who have HIV/AIDS. Before HIV/AIDS drugs were developed, most of the people who contracted AIDS died of fungal pneumonia because their bodies were not able to fight off the disease. Now, with the drugs, even though their immune systems are compromised, they don't have the possibility, at least not as great, of dying of fungal pneumonia. So these are some examples of diseases. Now, fungal diseases are very hard to treat. And the reason they're very hard to treat is shown by this particular branch over here. That is, we and the fungi share a tremendous amount of our biochemistry. We have a large number of similar pathways which we share with them. And so if we want to develop drugs to try and counter the fungus, we have to develop a drug which treats a fungal pathway and not a pathway that we have also operating in us. So in order to develop a drug to counter fungal diseases, you have to know two things. First of all, you have to know the identity of the organism that's parasitizing you. And then secondly, you have to know something about its biochemistry and how it differs from us. And in this regard, one of the major differences in the pathway or the biochemistry of the fungi and the humans is in the composition of the membrane. Our membranes contain cholesterol. Fungal membranes contain the compound ergosterol. And so in this particular example, when a drug has been developed to counter fungal infections, it has been targeted toward ergosterol synthesis, which is a pathway that we as humans don't have operating in us, and hence are able to use this as a drug and not have to worry about whether or not it's going to hurt us. In the last few minutes, I just want to show you a couple of slides indicating to you, let's see how we put the lights down here. We got the lights off. Somehow, the speaker has gone off. Can you guys hear me? Okay, now we're back on again. All right, well, I'm not sure how the lights, but we'll just do it this way. This is a fungus which grows in the area here, and again illustrates one of the generalities of fungi, that they're often brightly colored and that they're growing on organisms which are dead. Here is a fungus which parasitizes trees, secretes enzymes into the bark, and decays the tree. Here's another very yellow fungus. Yellow is a very prominent color in fungi, and you might ask yourself, why are these organisms so brightly colored? Who are they's attention are they trying to attract, and why do they want to attract that attention? Here is a fungus which grows around the Bay Area here. Here's a fountain pen, just to indicate to you a relative size. This is a fungus known as a chanterelle, and if you go to Berkeley Bowl, you can see that these are sold for quite expensive prices for food. Another very prized fungus which you can find here in the Bay Area is morels. These are very highly prized, very tasty. Here is the death angel, Amanita. This grows right outside the building here. And if you guys are careful and you walk past it, just look at it, but don't do anything else to it. Here is a fungus that parasitizes us. This is a fungus that causes some of the skin disorders for ringworm. For those of you who've had ringworm, this is what you will see, where the fungus is growing on your skin. This is a warning. The next slide is a little gross, but for those of you who want to be doctors, this is toenail fungus, a fungus which is very difficult to treat. And if you are taking drugs to treat this fungus, you have to have your liver monitored because the drugs also affect your liver function. Finally, I want to just show you a few common problems. Fungi decaying doors. And a picture taken right outside VLSB in the eucalyptus grove. Here's a eucalyptus tree, which was parasitized by a fungus, and when the wind came, it fell and knocked over the lamp. Here is a fungal disease which parasitizes plants. It's called rust. And finally, for those of you who are from Hispanic cultures or from South America, this is a fungus which parasitizes corn, takes the kernels and turns them into giant sorts of structures, which most of us wouldn't eat. But this is roasted in Mexico, and it's considered to be a highly delicious, very prized food. Okay, we'll continue talking about the fungi on Friday.